Hello, myself Harmandeep Kaur. I am the assistant professor of City University in the School of Agriculture and Natural Science. So today we'll discuss about the bacteria and its structure. We'll firstly start with the learning objectives. So with the help of agriculture microbiology, we people are able to study the nutrient cycling, soil fertility and organic matter decomposition. So in the soil fertility, we people are able to know how the bacteria are helpful to enhance the fertility in the soil, and in the decomposition we can study how the bacteria are able to degrade the complex molecules into the simpler one by using the various kind of enzymes. Moreover, plants and bacteria's relationships both are beneficial to enhance the growth of plants. How? For example, in the mycorrhiza or in the symbiotic association, both will combine together, with the help of which the bacteria provides the nutrients to the plants, and nutrients in lieu to above will enhance the growth of the plants. Thirdly, the microbial inoculants and biofertilizers can be used with the help of bacteria. For example, azolla. We people can use to enhance the fertility, which can be used in the powder form. Moreover, in the agriculture microbiology, we people can use the bacteria in the biotechnological processes. For example, we can transfer the plasmids from one bacteria to another bacteria. The live example is BT cotton, where the plasmid has been transferred from bacteria to just increase the productivity of the cotton, or to protect the cotton from the unwanted attack of the microorganisms, or you can say bollworms. Now let's start with the bacterial introduction. So bacteria is a single-celled microorganism which is prokaryotic in nature. Prokaryotes are those which we can't see with the naked eyes. So they are of tiny structures, and always we people use the microscopes to do the study. Moreover, the bacteria are ubiquitous in nature, means they are present everywhere, in the soil, in the air or in the water. So the bacteria don't have the two nucleus, so membrane bound organelles are absent in the bacteria. So we people can find the bacteria in different kind of environments, for example in the harsh conditions or in the normal conditions. So those bacteria which are present in the harsh conditions, such as in the salty areas, those bacteria are known as halophiles. Moreover, which are present in the acidic soils, they are known as the acidophiles. On the basis of their availability in the different conditions, we can divide the bacteria into different kinds. So bacteria can be used in the nutrient recycling. Nutrient recycling means in the environment carbon dioxide is there, oxygen, sulfur, nitrogen, all are present. So with the help of bacteria, different kind of enzymes will be produced which convert the complex structures into the simpler one. So what are the uses of bacteria? So firstly, pest control. As we people have already discussed, Bacillus thuringiensis, it's a kind of bacteria which is used to protect the cotton from the bollworm. Moreover, in the bioremediation, we people can use the bacteria to decrease the pollutant or pollution in the environment. Furthermore, the bacteria can be used in the fermentation, means for example we people daily eat the curd where the lactobacillus bacteria is there. So the same bacteria can be used for the different kind of processes related to the fermentation. So now let's start the structure of bacterial cell. So firstly, the outer covering of bacterial cell is known as the cell envelope, which consists of cell wall and cell membrane. Cell wall is not too much rigid. It is made up of peptidoglycans and also contain the sugars and amino acids, which provides the sticky nature to the bacteria. Furthermore, below the cell wall, cell membrane is present. So cell membrane basically enclose the cytoplasm of bacteria. Cytoplasm is the semi-liquid kind of organelle, or you can say part of the bacteria, which is present at the center. So with the help of cell membrane, there are some pores which are present in the bacteria, which allows the movement of liquid, movement of water from inside to the outside of the bacterial cell. Next, start with the cytoplasm. As I have already discussed, is a semi-liquid part of the bacteria which contain different kind of organelles. Third, nucleoid. Nucleoid is the irregular shape region which is present in the cytoplasm, which contains the genetic material of the bacteria. So, as the bacteria is prokaryotic in nature, so it doesn't contain any kind of membrane bound organelle. Next is plasmid. So, as we have discussed about the use of bacteria in the biotechnology, so mainly the plasmids are helpful to transfer the genetic material of one bacteria into the another. Next, ribosomes. So basically ribosomes are helpful to enhance the protein productions in the bacterial body. With the protein production, there will be the enhancement of the bacterial growth. Furthermore, flagella. Flagella is the tiny thread-like structure which is helpful for the bacteria to move from one side to another side. So on the basis of bacterial types, the flagella is of different kind. We can divide it into the peritrichous, monotrichous, lophotrichous, amphitrichous, which allows the movement of bacteria from one side to another side or into the different directions. Next is pili. It's also known as the fimbriae. So pili is also third name is conjugation tube, which allows the exchange of genetic material between two bacterial bodies. So it helpful for the conjugation process by making the conjugation tube. This is the bacterial diagram as we have discussed. So the cell wall is there, below the cell wall plasma membrane is there, which contains the cytoplasm at the center of the body. So nucleotide is present there, which is made up of red kind of structure. So flagella is helpful, as I have told you, for the movement of the bacterial body. So all are the organelles or the part of bacterial structure. Next, flagella types. Firstly, flagella is also known as the monotrichous. If only one flagella is present on the bacterial body, so we can call the bacterial body as monotrichous. Second is peritrichous. So a lot of flagellas are present all over the bacterial body, which allows the movement of bacteria into the various kind of directions, like from unidirectional to bidirectional or in any kind of direction. Next is lophotrichous. If the bunches of flagellas are present only on the one side of the bacteria, then those kind of flagellas are known as the lophotrichous. So bacteria containing the bunch of the flagella will move towards a specific direction, not in all directions. These are the structures showing the different kind of flagellas on the bacterial body. Firstly, peritrichous, all the body is covered with the flagella. Atrichous, no flagella is there. Monotrichous, only one flagellal body is there. Amphitrichous, two kind of flagellas are present on two different directions. Lophotrichous, bunch of flagella is present. Next, amphitrichous. As I have told you, so in the amphitrichous, the flagellas are present on the both direction. It means the bacterial can move bidirectionally. Atrichous, when there is no involvement or presence of the flagella on the bacterial body. So mostly these kind of bacteria are non-motile, means they can't move from one to another. Next, how the bacteria are able to reproduce or divide. So firstly, main mode of reproduction is conjugation. So conjugation allows the genetic transfer of plasmid from one bacterial body to a second bacterial body. So mainly two kind of bacterial bodies are there. Firstly known as the donor, which gives the plasmid to the recipient, and we always indicate the donor with F positive and recipient with the F negative. So here firstly let's introduce the process of conjugation. Firstly, donor cell is also known as the F positive, because F positive indicates the presence of plasmid as well as bacterial chromosome in the bacterial body. Second, when one bacteria sends the another bacteria in the surroundings, then it sends a signal which allows the formation of pilus. Pilus means it's a kind of conjugation tube with the help of which the plasmid will move from one body to another body. So after the connection between two bacterial bodies, the F positive transfer its plasmid from F positive to F negative, and at the last the recipient again gets converted into the F positive. So this is the diagram which showing the conjugation. In the first diagram we people are able to see the donor cell, which is indicated as F positive, and the recipient cell, which is indicated as the F negative cell. So sex pilus is there, which is known as the pili. Okay, and I have already told you the names of pili is fimbriae or conjugation tube. So a circular body of blue color indicates the F plasmid, which will be transferred from F positive strain to the F negative strain. So in this second structure, you people are able to see like the conjugation tube is formed between two kind of bacteria, and in the third, the plasmid is going to transferred from F positive to F negative, and at last you people are able to see the recipient cell, which was F negative earlier, gets converted into the F positive, means the exchange of genetic material is done and F negative strain becomes the F positive. So this is all about the bacterial cell and the introduction of the prokaryotes. I hope you have enjoyed the lecture. Thank you.